Brooke Jones, who received her PhD from the USM at the Stennis Space Center, and she currently has a postdoctoral research associate there. She's the project lead for the Dynamic Anomaly Project at USM's Ocean Weather Lab and is a member of the Concord Consortium, and her research focuses on biogeochemical impacts of land-ocean interactions and the development of novel marine surface products derived from satellite observations and ocean models. And she's going to go ahead and give us a good explanation on the Ocean Weather Lab and all the great technology they use there.
Okay, so thank you for having me, and then everyone for staying to listen. I think I'm the last talk of the day. So as Monica was saying, I work at the Ocean Weather Laboratory, which is one of the research laboratories that USM operates at Stennis Space Center. And one of our tasks one of the objectives that we're tasked with in our laboratory is what we call continuous monitoring of the Gulf of Mexico. So that's a little bit of what I'll present today. Our mission in the Ocean Laboratory is this maybe somewhat complex sounding sentence, which is we want to assemble and couple satellite remote sensing data with ocean models, in situ observations, other remote sensing products to fully characterize a four-dimensional ocean. So what we really mean by that is that we want to be able to implement and integrate different types of data across different scales that we can retrieve with satellites, that we can model, and that we can get with shipboard and field-based data. And we want to understand the ocean not just at one point, but how properties of the ocean are distributed laterally, zonally, with depth, and then over time. So that's the four-dimensional idea of the ocean. And how we can apply this in our laboratory, that's just a picture, because I actually thought that was kind of neat. The first time I walked in, it looked like some type of operation room, which is a big difference from the research I'd been doing. So anyway, I like that picture. But so what I will go through today about how this can relate to oil spills is, what do we mean when we say ocean weather? How can we measure the weather of the ocean? What do we do with that information? And then how is it applied to oil spills? And the last two are really kind of the same for this talk. So when we think of weather, most of us think of atmospheric weather, the conditions of the atmosphere. And we're used to seeing satellites and models that forecast how that weather is going to change, what we can expect on a daily basis or several days in the future. And we don't often, unless we are oceanographers or working in marine science, think about the fact that the ocean is actually very similar to the atmosphere in that it is a fluid body that's under the same types of controls of heat, advection, movement, and currents that we would expect to see in the atmosphere and with that type of weather. So when we say ocean weather, what we're referring to are ocean conditions at any given point in time or forecasted into the future. We use the same types of techniques as you would expect to see in atmospheric weather. And in our laboratory, what we're really interested in is how can we look at the physical characteristics of this four-dimensional ocean and be able to explain something of what's happening with the ecosystem. So we're very interested in how water moves, how heat and energy are transferred through the ocean, and how marine organisms respond to that type of condition. So that's really what our weather lab is exploring. The way that we measure this is very similar to atmospheric weather. So we use ocean satellites. That's probably our primary data source as far as true observed data. And if you're not familiar, there are multiple ocean satellites at any time, public ocean satellites that have data available for anyone to use and to download, and we take advantage of that. The satellites have been in operation since 1978. Obviously not the same satellites now, there's been quite a bit of evolution. But at any time we are continuously monitoring temperature, surface height of the ocean, and ocean color. So ocean color is where a lot of my research is focused, and that really gets into the biogeochemical and biological response of the ocean by looking at how radiance or how light is reflected back to satellites and what that can tell us about what's in the water. So we can use this kind of information to judge water quality. We can look at really interesting features like river plume boundaries, so where the Mississippi River is this fresh, nutrient-rich water that's entering into the salty Gulf of Mexico, we can actually trace that using ocean color amongst other types of conditions. One thing that we're constantly trying to understand are algal blooms and so that's how phytoplankton bloom and grow and then eventually decay. These can be harmful algal blooms, and they can also, so they can present a danger to marine life, and they can present a danger to humans at times. And then, of course, finally, we can look at impacts from events, like something like an oil spill or a flooding event, and ocean color can give us a sense of the impacts of those events. So for ocean color, we haven't had continuous data since 1978 like we have with some of the more physical parameters. We actually started measuring ocean color in the 1980s, but we didn't develop a continuous data set until 1998, and that was with the CWIF sensor. We have maintained relatively stable. There's been differences as far as our technology goes in what we can measure, the resolution that we can measure. And right now, our sort of state-of-the-art ocean color measurement device is a sensor aboard polar orbiting satellites called the VIR sensor. So this VIR sensor measures ocean color with a very advanced resolution of 750 meters, which to a satellite oceanographer sounds like really great resolution. But if you think about how large that is, that's several football fields, then we're trying to study something as small as particulates in the ocean that might be affecting ocean color. It doesn't even sound like we can really do that. And as far as being able to look at something like what Adam was showing earlier, and these interactions of these small particles, we really can't. What we can see are large-scale processes. So luckily for us, ocean color and the particles, like for instance phytoplankton that contribute to ocean color, tend to be in very large concentrations, or their lack of concentration is also indicative of water quality. So while we're not able to resolve down to individual species or individual population members, we can look at how the phytoplankton distribution changes with respect to the conditions or the weather of the ocean. And I just threw the last part in there just to give you an idea of how much data, why we would even bother with looking at satellite data if when we have this large resolution, is because just for one satellite, just one day on one sensor passing across the Gulf of Mexico, we get over 6 million data points. So we have a continuous record now for 20 years of nearly 6 million data points per sensor every day, which means that we can look at any location across the Gulf of Mexico and have a nice time series and understand how these processes occur over time. So, oh wait, I thought I was going to talk about models. Okay, this is just two examples of some of the satellite imagery that we use, and so I wanted to show how this is ocean color. Both of these images are chlorophyll, but they're taken on different days. They're actually several weeks apart. That's why there's considerable difference between the patterns that you see. So the darker colors, or the warmer colors, are going to be higher concentrations of phytoplankton, and we see that closer to shore. This brown is the shore. I don't know if that's really obvious that this is Louisiana going over into Florida. And the lower concentrations of phytoplankton that we see out here, we associate there's probably something physical happening that's causing not just a lower concentration than what we have in coastal areas, but we can actually see that there's something, perhaps a feature itself, that's causing a lower concentration here. And those are the types of things that we're interested in understanding and researching. So every day we are mapping these types of products from the VIR satellite, and we do this for 16 different biooptical or ocean color properties. Okay, so here's the models. We also, to better understand the physics of the ocean, we look at some of the Navy models. So we take 3-hour increments from the Navy models, and we are using two of them. Their operational model, which is HICOM, and one of their local or regional research models, which is AMSES. And we use these to try to understand how the ocean is moving and how temperatures and heat are fluctuating throughout the ocean. not just in response to those biological features, but also at depth. So one limitation that we have from the satellites, aside from the resolution, is that they typically only can see satellite or surface patterns. The models allow us to calculate, given some known and estimated equations, how something like currents, temperature, salinity, how those features propagate at depth. And that allows us to understand more of the mechanisms that are driving what we see in surface properties. They are, at least as far as the readily available information, they are even rougher resolution than what we see with the satellite. So it's up to 3 kilometers resolution, at least for what they put out daily. So that can be varied for specific studies, but that's yet another limit that we have with models. And here are 2 corresponding plots with the satellite data that I showed a minute ago. These are of current magnitude. So these are both coming from the AMSES model. And you can think of current magnitude as being the strength of the current. You can see the strong current that seems in the same location as where I was indicating that there was low chlorophyll earlier. We see this strong pattern. This is actually, we know that this is where an eddy is separating from the main body of the loop current, which drives most of the circulation within the Gulf of Mexico. Okay, so what can we do with this? And driving this, so at the end I'll say how we can look at events like oil spills. But with driving this explanation today, we want to understand when we have something like an oil spill in the northern Gulf of Mexico, how is that going to impact known processes? If we see something like a spike in chlorophyll, how will we know whether or not that spike is either directly related to the oil spill or even falls out of the normal range of what we'd see during that time of year? So part of how we want to understand this is by looking at, by visualizing these types of features overlaid with each other so we can see what the controlling forces are, and then also look at the long-term time series to understand these fluctuations. So this is Google Earth. I decided not to actually try to open Google Earth and see if it would run in here today. So these are just snapshots taken from Google Earth. You can see the bathymetry that's from Google Earth itself. That's not something that we calculated. And I'm first going to overlay this with a chlorophyll image, which is happening here. But, here it is. Okay, so this is just south of Texas. And you can see again, like we saw in the earlier image, there's higher chlorophyll near the coastline. And we see it's still relatively elevated as we move offshore, and then significantly decreases. This is around the shelf edge. So as we move off the shelf of the water, we start to see a significant decrease in the amount of phytoplankton. And that's what we would expect to see. We do, however, see this kind of swirled pattern of chlorophyll here. And when we take our model data and overlay the current vectors, I'm not sure what's happening here, but something just shut down. Okay. When we overlay the current vectors, we can look to see whether or not this is offshore advection of water or something else that's maybe leading to these patterns that we see of this off-shelf higher concentration of chlorophyll. And we do, in fact, see that there is this southeastward current-driven process that's occurring, so that's likely linked to what we're seeing here in the chlorophyll signal. We also, if you remember, I pointed out the loop current. This is part of that eddy that we were looking at just a moment ago. You can start to see this almost like anticyclonic rotation here in the current vectors, and that's really what leads to that low chlorophyll signal that's associated with the loop current. So one interesting thing that we can do, like I said, we want to look at the time series. What we have with this continuous monitoring, one of the advantages that it gives us is that at any point we don't have to be reactive to something like an event. We have a time series at any point across the Gulf of Mexico for all of these parameters. So we're looking at around 16 satellite parameters and around 12 model parameters that we're processing every day. So if we have a region of interest, be it for an event or just simply something that maybe someone has a station that they're interested in the results that they got when they were out on a cruise, a research cruise, we can pull up the time history for any location and look to see how parameters in that area evolve over time. So for this offshore area, and we are off shelf here, you see a really smooth, nice seasonal pattern of the parameters that are featured here, and I don't even know what these parameters are, but that's not necessarily the point here. If we then look at something that's more near shore, we see that the same set of parameters over time appear much more chaotic. So this area is going to be influenced by more episodic events like runoff from land. There's going to be a difference in circulation features. And while we do over time see a seasonal pattern, it's often perturbed by these episodic events. So there's a real difference here, and you can imagine a real difference for how an event like an oil spill might affect these two very different regions, despite the fact that they're both considered in the northwest Gulf of Mexico. Okay, so more specifically, how does this apply to oil spills? Well, in some ways we don't know, because fortunately and unfortunately we have not had another oil spill in this area that we could study since we began this project. We do have other events that we can look at and use as a bit of a proof of concept of how we can integrate these satellite and physical models to understand processes and events in this area. So this is that same swirl pattern. I just made a smaller area here. This is the daily image of chlorophyll that we just looked at. And in this black box, I've delineated where the Flower Garden Banks coral reefs are south of Galveston, Texas. This time period was when a mortality event, a very significant mortality event, was discovered at that Flower Garden Banks. And the reasons for the event were unknown, but it was thought to be linked to coastal flooding that somehow moved out over the coral reefs. So what we did was we compared our chlorophyll signal to the typical signal of chlorophyll for that month in our time history. And visually, when we look at this, we don't really see any difference. I mean, to me, and I look at these every day, those look like basically the same color. That looks like the same ocean color. So we looked, wanted to take a bit more quantitative approach to this, and we looked at the difference between the two to see how normal or abnormal were these conditions during the day that this event was discovered from this typical image. And it may be a little bit difficult to see with this color scale, but there's a slight elevation here in absolute value of chlorophyll. This is another type of difference field that we can calculate, which is how different this daily image was from the previous conditions, so how things had been evolving in the last 2 months rather than for the entire time series. But regardless, even when we look at this, when we visually look at this abnormal change, we're not really seeing that there's anything that really strikes us as, oh, well, look, this is really a hot spot, something was occurring here. However, when we take that abnormal plot and we turn it into its own time series, so what that means is we're basically computing an anomaly. So we take the entire time series from January of 2014 to present at this time, which was January of 2018, and we plot every time that the chlorophyll values in that area were higher than normal, and normal being this 0 line, and every time that they were lower than normal, we start to see peaks in, and we start to see patterns within this abnormality range. So the peak that we're interested in, I tried to note with this blue line, this was the actual date that they discovered the mortality, so they're interested in what led up to that period. Each of these highlights represents peaks that occurred over the entire history of this area, including the peak of interest for us that fell out of the normal standard deviation. So it was in fact a significant peak in chlorophyll, which we're using as a proxy for turbidity here, or how cloudy the water was, how much light was making it through the water. However, it's not the highest peak by far, even though it led to this really significant die-off event. But the other thing that we could look at was how long each of these significant peaks persisted in the environment. So this was over the entire 4-year history that we have with this satellite, the highest or the longest lasting period of high chlorophyll or positive anomaly that we saw in the region. So while that doesn't necessarily tell us what led to the event, it gives us some historical analysis for which to compare it. And we'd like to be able to take this type of analysis in the event of an oil spill and be able to look at any effects that the oil spill would have on the local ecosystem by understanding better what the processes are, what the seasonal patterns are, and what type of interannual variability we see, like we can do with this coral reef problem. And I actually think that I left out my summary slide, so that's it.
Thank you.